All right, welcome everyone. Today we're going to talk about the structure and function of large biological molecules or macromolecules. Our objectives with this lecture are to demonstrate an understanding of the basic chemistry of life. We'll talk about the four major groups of organic molecules associated with living things. We'll be able to describe the general function of each major group, factors affecting enzyme activity, and you'll also be able to explain the difference between dehydration synthesis and hydrolysis. So, to start off, all four of the biological macromolecules are made up of monomers, which are small individual units for building. You can think of these kind of like beads on a string or like individual Legos. Polymers are longer molecules made up of monomers. They may have many identical or similar blocks linked together by covalent chemical bonds. So monomers, two monomers are connected together to build a polymer by a special reaction called dehydration synthesis or a condensation reaction. Water's produced in this type of reaction. Polymers together are connected to create macromolecules. So these are large functional molecules that have the ability to do some sort of job in a living organism. So monomers are the individual units. They're tiny. They're linked together by dehydration synthesis, a special chemical bond that when it's formed releases water. All right, so here we go. We've got dehydration synthesis. So this is how polymers are made. We have many monomers, so like two different monomers, we might say A and B, combined together to form polymer AB. So this is something where this polymer has different characteristics than each of the individual monomers do. And by dehydration synthesis, we see A plus B equals AB plus water. This is the dehydration synthesis general formula. Hydrolysis is the opposite. So if you're going to break down a polymer into its individual monomer units, you have to have water present to help make that happen. And that's where this is called from. The name comes from hydro, meaning water, and lysis is to break apart. So we're going to hydrolyze the polymer AB in the presence of water and break it down into individual monomers, A plus B. You're going to need to know dehydration synthesis and hydrolysis in its general forms. You should also know, and by the end of this lecture, it should be clear what types of polymers are created from what types of monomers. All right, here's another example of a dehydration synthesis reaction. So if we have a short polymer with a hydrogen on one end, and we want to link it to another monomer that has a hydroxyl group, an OH group, you see we have two H's and an O. When those two come together, they leave as water. And then these two bonds combine together to link the two monomers together. So we end up with a polymer that's now got 4 monomers on it and a free water. Now to do the opposite, to remove a monomer, we add a water, and that puts a hydroxyl group on one end when this bond is broken. The monomer on the right gets a hydroxyl, and the monomer on the left gets a hydrogen. And here's another way to visualize it. You can see here where the OH and the H are in these two amino acids, which are the monomers that make up a protein. So if we combine these two to produce a water, that leaves a place here for a bond to form between this carbon and this nitrogen. Carbon and nitrogen right here. And the name of that type of bond in a protein is called a peptide bond. Here we see the same thing in a carbohydrate. We've got one monosaccharide, which is the monomer for carbohydrates. One monosaccharide here, a second monosaccharide there. we see an OH and an H. These can leave as water in a dehydration synthesis reaction, and that leaves these two connected by that shared oxygen in the center. All right, here's an example of hydrolysis. So we're breaking apart a polymer to produce monomers. So we've got a triglyceride, a fat here. In the presence of 3 waters, it'll break this down into glycerol and fatty acids. But water has to be present for that to occur. All right, so what is the relationship between dehydration synthesis and hydrolysis? I'd like you to pause the video and write it down in your own words. How are these two chemical reactions related to one another? Okay, next, I'd like you to start a spot in your notes where you're tracking what are the 4 types of biological macromolecules. I mentioned a few already, but we'll go through them each one by one. First, we'll talk about carbohydrates. Carbohydrates are used as fuel and building material by living organisms. They include simple sugars like fructose and polymers like starch, which are larger and more complex. They have a general ratio of 1 carbon to 2 hydrogens and 1 oxygen, or CH2O, which is where they get their names, carbo for the carbon, hydrate for water, because of the ratio of these 3 elements. The monomer for carbohydrate is a monosaccharide. 2 monosaccharides together are called a disaccharide. The prefix di means 2. And many monosaccharides together produce a polysaccharide. Monosaccharides are the monomers. These would be things like glucose or ribose. Polysaccharides are larger molecules like starch or glycogen for storage of energy. They could also be structural, like cellulose in plants. That's the fibrous stuff that gives plants their structure and their ability to stand up. It's what wood is made of. There are also some animals that produce polysaccharides that are structural, like chitin. And chitin would be like the stuff that makes up the exoskeleton of a beetle. Okay, here's some different types of structures and classifications of monosaccharides. They're usually named for the number of carbons they have. So a 5-carbon sugar might be some kind of pentose. A triose would have, you guessed it, 3 carbons. And hexoses would have 6 carbons in them. There are linear and ring forms of sugars, and it's the same molecule. It just depends on whether it's dry or it's in an aqueous or a water solution. Generally, these sugars are linear when they're dry. Think of like the granulated sugar in your kitchen at home. That would be a linear structure. But when you dissolve it in water, these ends snap together and form a ring, which is what you see here. And that ring structure is what we see predominantly in a water-based solution. All right, so here's an example of a dehydration reaction in the carbohydrate, linking this glucose to another glucose to produce a disaccharide, maltose. We have an OH and an H. They leave together to form water, and that bond snaps together here between that leftover oxygen. And we also have another example here with sucrose, a dehydration synthesis reaction. Same thing, the OH and H leave together as water, leaving an oxygen in the middle to share that. And the name for this type of bond that connects monosaccharides together in a polysaccharide is called a glycosidic linkage. All right, there are 2 forms of glucose. There's alpha glucose and beta glucose. And what it's got to do with is just the position of the hydroxyl group. See where this one's opposite the hydroxyl group here, and this one it's on the same side. Now in starches, it uses alpha glucose monomers. See how the hydroxyl groups are all on the same side? In cellulose, beta glucose monomers are what's used. So we see they alternate here. All right, and let's see where these polysaccharides are actually located in living cells. Oops, sorry, wrong way. All right, at the top here we have a picture of plant cells. So this is a chloroplast, and these big white structures are starch granules. Starch is usually made up of amylose, amylopectin, and these are energy storage molecules to be used on a rainy day. Someday when it's cloudy and this plant can't do photosynthesis to produce glucose, it can access the energy stored in the starches here, in the starch granules. In an animal cell, we store energy for later use as glycogen. And these glycogen granules just hang out in the cell until they're needed for a day when we can't find food to digest and break down. We have this backup storage energy of glycogen granules within some of our cells. All right, I mentioned chitin earlier. Here's a good visual example of the exoskeleton of an insect. These are made up of chitin, a structural polysaccharide. All right, so take a moment here, pause the video, and describe the structure of simple sugars with 3 to 6 carbons. Remember, they're called carbohydrates. So if there's 3 carbons, how many hydrogens and oxygens would you expect to see? Next, I'd like you to relate the structure of polysaccharides to their functions. The simple sugars are used for energy. The longer, more complex molecules with all those energy-rich bonds are used for energy storage for a later day. All right, the next type of macromolecule we're going to talk about are nucleic acids. Nucleic acids' job is to store genetic information. And there are 2 different types of nucleic acids. We have DNA and RNA. And DNA is double-stranded in a helix. It's made up of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. It stores hereditary information, and it is a very long, large molecule. The sugar that makes up DNA is deoxyribose. Remember, DNA stands for deoxyribonucleic acid. RNA, on the other hand, is a single-stranded molecule. It's usually temporary. It's made up of 4 nitrogenous bases too, except that instead of thymine like you'd find in DNA, RNA uses uracil. So if you ever see a T in a sequence, you know you're looking at a DNA sequence. And if you see a U, you know you must be looking at an RNA. RNA carries information from the DNA to the ribosomes, and it's involved in transcription and translation, which we'll get to at a later chapter. But it's enough to know now that RNA is important for going from DNA to actually producing proteins at the ribosome. The sugar that's used in RNA structure is ribose, which is why RNA starts with R, ribonucleic acid. Nucleotides are the monomers of DNA and RNA, and a nucleotide is made up of a nitrogenous base, so A, T, G, and C in DNA, or A, U, G, and C in RNA. They'll have a sugar, a 5-carbon sugar, pentose. It could be deoxyribose or it could be ribose. depending on whether it's DNA or RNA. And it also always has a phosphate group. And the phosphate and sugar make up the backbone of a nucleic acid molecule. So you've got our 5-carbon sugar with its phosphate group and a nitrogenous base. And these are the 3 components that make up nucleotides. All right, the purines are the amino acids adenine and guanine, and they always bind one purine to one pyrimidine, and in very predictable fashion. A binds with T, G binds with C. Now in RNA, T's are swapped out for U, so A can also bind with uracil. Purines have a double ring structure. Pyrimidines have a single ring structure. Take a moment here, pause, and try from memory to, describe the structure of a nucleotide. You could even draw a diagram like one on the previous slide and label the parts. All right, so here's some pictures of our nitrogenous bases. We've got the pyrimidines with just one ring on top, cytosine, thymine, and uracil. Purines on the bottom have a double ring structure, and they always bind A with T or A with U if it's RNA, and G binds with C. One trick you can use to remember that is apples and trees, cars and garage. All right, compare and contrast the structures of DNA and RNA. How do they differ? How are they similar? When you're ready, move on to the next slide. All right, so there's a flow of information in a cell from DNA, which resides in the nucleus, and it's trapped within the nucleus. RNA is able to be formed in the nucleus, but it can exit the nucleus and travel through the cell to the ribosomes, where the information in that RNA molecule is actually used to create a protein. Here you can see the monomers for amino acids building up a polymer, a polypeptide here at the ribosome. Pause this video and describe the function of DNA and RNA quickly. You should also be able to recognize other nucleotides involved in energy metabolism. All right, now let's move on to proteins. Proteins, or proteos, comes from Greek for first or primary, and they're 50% of the dry weight of cells. So if you were to put, even think of like a piece of steak in a dehydrator to make jerky, 50% of the jerky's weight is going to be protein. All right, this contains, proteins are made up of carbons, hydrogens, oxygen, nitrogen, and sulfur. Now, sulfur is an element we haven't seen come up yet in our discussion of macromolecules. And here is a model of what myoglobin protein actually looks like. All right, some protein functions. We've got many proteins are biological enzymes. Enzymes are a catalyst that makes reactions happen faster and require less energy than they would normally. So lactase is the enzyme that helps break down the sugar lactose. Would that be a dehydration synthesis or a hydrolysis reaction? Breaking down lactose sugar. Good. It would be a hydrolysis reaction because you're breaking apart a more complicated molecule into simpler pieces. Good. Proteins are also really important in defense. They make up our antibodies. Storage. So milk protein, casein, is a way to store energy for later. They're important in transport. So hemoglobin is the protein that transports oxygen to your cells and also carries away carbon dioxide. Proteins also make up our hormones, things like insulin and receptors. They're also important for movement. Some cells have motor proteins like flagella or cilia. These are important structures. Some other structures can be things like keratin, which is what your hair and fingernails are made up of. All right, so an overview of protein functions. We have enzymatic proteins. They do selective acceleration of chemical reactions. For example, digestive enzymes catalyze the hydrolysis of bonds in food molecules. We have defensive proteins, and these function against disease. Antibodies inactivate and help destroy viruses and bacteria. Storage proteins are used for storing amino acids. For example, casein, the protein in milk, is the major source of amino acids for baby animals. Plants have storage proteins in their seeds. Ovalbumin is the protein of an egg white that's used as the amino acid source for a developing embryo. And transport proteins, they move substances into and out of cells. For example, hemoglobin is the iron-containing protein in vertebrate blood. It transports oxygen from the lungs to other parts of the body, and other proteins also transport molecules across cell membranes. There are hormonal proteins as well. These function to coordinate an organism's activities. For example, insulin is a hormone secreted by the pancreas, and it causes other tissues to take up glucose, thus regulating blood sugar concentrations. Receptor proteins, these function to mediate the response of a cell to chemical stimuli. For example, receptors built into the membrane of a nerve cell can detect signaling molecules released by other nerve cells, so they can pass a message along from one nerve cell to the next. We also have contractile and motor proteins. These are important for movement. Motor proteins are responsible for undulations of cilia and flagella. Actin and myosin proteins are responsible for the contractions of muscles. And finally, structural proteins. These provide support. Keratin is the protein in hair, horns, feather, and other skin appendages. Insects and spiders use silk fibers to make cocoons and webs, respectively. And collagen and elastin proteins provide a fibrous framework for animal connective tissues. There are 4 levels to protein structure. Primary structure, this is the amino acid sequence. So kind of like beads on a string, which amino acids need to go together in which order to produce a functional protein. That's the primary structure. There are 20 different amino acids in total. Peptide bonds are the name of the bonds that link together each pair of amino acids. Amino acids have a structure like this. They all have an amino group with this nitrogen. They all have an alpha carbon and a carboxyl group. This is a carboxylic acid. So you have amino and an acid, hence the name. Now, the thing that differs between each amino acid is the side chain, and that's what this R group here. R in this instance is a variable. That's a placeholder like X in algebra. It is not representative of radon or some other element that we represent with a single letter. So this R is just a side chain, some other group. So the R group can be hydrophobic or hydrophilic. So it can be water-fearing or water-loving, and that plays a big role in the behavior of the amino acid. It could also be ionic, like an acid or a base. All right, so here we see amino acids separated by ones that are hydrophobic are all up top here in yellow. Hydrophilic ones that have polar side chains are here in the middle. and ones that have an electrically charged side chain, these are all hydrophilic, by the way, would be acidic and basic ones over here. Good, okay. So 2 amino acids are held together by a peptide bond. That bond is formed by dehydration synthesis, so there's our water leaving, and that's going to let this carbon connect to the neighboring nitrogen here and form a peptide bond. You'll need to know the names of the monomers, amino acids, the type of bond that's formed, peptide bond. It's formed by the process of dehydration synthesis, and the product is a protein, or sometimes we talk about polypeptides, would be a long string of amino acids, but not yet a fully functional polypeptide protein. All right, the secondary level is when the protein begins to gain its 3D shape. It has folds and coils caused by hydrogen bonding between those side chain groups. We have alpha helix, where the protein twists itself into a helix like this. And we also have something called a beta pleated sheet, which kind of looks like when you're a kid and you fold up a sheet of paper to make a fan. That same kind of structure. Alpha helix, beta pleated sheet. These are secondary structure elements. The basic principles of protein folding are that hydrophobic amino acids are buried in the center of the protein, and hydrophilic ones are exposed on the surface, right? Now, most of life occurs in water, which is why proteins are shaped this way. Acidic and basic amino acids form salt bridges or ionic bonds, and cysteines can form disulfide bonds. A cysteine is one type of amino acid. All right. The third level, tertiary structure, is bonding between the side chains, or the R groups of amino acids. Hydrogen bonds, ionic bonds, disulfide bridges, and even van der Waals interactions produce these types of structure, tertiary structure. You don't need to stress about the different types of chemical bonds here. Just know that tertiary structure occurs when there's bonding that happens between the side chains. All right, and lastly, quaternary structure is when 2 or more polypeptides bond together. So we can see in the example here, collagen is made up of, this one has 3 different polypeptides that are woven together. Or hemoglobin, here we've got 4 different polypeptides that are working together to create one functional hemoglobin molecule. All right, so take a moment while it's fresh in your mind. Describe the 4 levels of protein structure in your own words. All right, let's skip this question about motifs and domains. All right, what's the relationship between amino acid sequence and their 3-dimensional structure? So if you were to change the primary structure, right, the order of amino acids, would it impact their 3-dimensional structure at all? Yes or no, and why? All right, and here's a good picture to kind of show you them all. So the primary structure is just the order of amino acids. The secondary structure is where we see alpha helixes or beta pleated sheets being formed. The tertiary structure is how those side groups interact with each other, and that causes the hydrophobic parts of the protein to be on the inside, and the hydrophilic parts should be on the outside. And then quaternary structures when we put multiple polypeptides together to produce a functional protein. Chaperonins are, it's like a tiny cylinder. Polypeptides go in, a cap goes on and attaches, and it folds the protein in such a way that now it's functional. So the folding of proteins isn't random. It's not like just putting a bunch of magnets together and hoping all the positives and negatives line up. These chaperonins deliberately fold proteins in specific ways so that they're correctly folded and ready to go off and do their job. Protein structure and function are sensitive to chemical and physical conditions. It unfolds or denatures if pH and temperature are not optimal. So if you think back to our yogurt lab that we did the first day of class, regular milk that has not been treated with acid or temperature is very liquid, right? There's normal milk proteins in there. However, we denature those proteins by exposing it to extreme heat. Some of those proteins begin to denature and the milk thickened, right? And then as we let our yogurt culture and we let the bacteria break down the lactose sugar and produce lactic acid, the pH in your milk dropped. And that low acidic pH further broke apart those hydrogen bonds and made the milk thick, right? At that point, we had yogurt. So keep that in mind. I think making yogurt is a really good way to visualize denaturing proteins with both temperature and pH. All right, so a change in structure produces a change in function, right? Here we see a normal primary structure for normal hemoglobin, and it produces round red blood cells that are shaped appropriately to do their job and carry oxygen throughout the bloodstream. However, in people with sickle cell anemia, they have a change at the 6th amino acid. They have a valine instead of a glutamine. And a change of just that one amino acid changes the end result enough so we get these crescent moon-shaped cells, these sickle-shaped cells. These cells are not nearly as good at carrying oxygen through the bloodstream as a red blood cell that's round. So the implication or the consequence of this change of just a single amino acid is very severe. All right, how does our knowledge of protein structure help us to predict the function of unknown proteins? All right, think about this one. See if you can come up with it. All right, lipids are our last group of biological macromolecules. Lipids are fats, waxes, and oils. Now, fats are things like triglycerides, and these are used as another energy storage molecule. There's more energy, there's more calories in a gram of fat than there are in a gram of proteins or a gram of carbohydrates. So if you have to carry around extra energy, it's much more efficient to carry around fat than carbohydrates or proteins. A triglyceride is made up of 3 fatty acids and a glycerol. There are saturated, unsaturated, and polyunsaturated fats. Also within the lipid group are steroids, and these would include things like cholesterol and hormones. The next group are phospholipids. This is the lipid bilayer of your cell membranes. These are very important to functioning of organisms. They are made up of a hydrophilic head and hydrophobic tails. All right, so let's look at some examples of chemical structures. We've got a fatty acid up here and a glycerol. So one of 3 dehydration reactions in the synthesis of a fat molecule. So here we've got our H and our OH. These will leave together as water. And this oxygen will bind with this carbon here. And the name of this bond is called an ester linkage. Now, one thing that's different in producing a triglyceride is that this dehydration synthesis reaction has to happen 3 times. So the structure of a triglycerol always looks like a capital letter E. All right, take a moment, in your own words, describe the structure of a triglyceride. Okay, now I mentioned there's different types of fat, right? We have saturated fats, which are saturated with hydrogen. These are most commonly found in animal fats, things like lard and butter. And they have straight chains because each carbon has the maximum number of hydrogen bonds. So straight chains, saturated fats. And at room temperature, these fats are usually solid. All right, and compare that with unsaturated fat. Unsaturated fats are bent because there's some double bonds in here. Carbon to carbon double bonds that kink the chain. These are the types of fats we generally find in plants. They are liquid at room temperature. Because of these kinks, they don't settle nicely into a solid shape. At room temperature, they need to be colder. Examples of unsaturated fats would be things like corn oil and olive oil. Now here's a peek at cholesterol. Notice the rings. These are different, very different in shape than what you'd see in a triglyceride, right? So if the structure is very different, what do you think about the function? Is the function likely to be different too? Yeah, good. Okay, and then here we are looking at a phospholipid. These are the types of lipids that work together to make our cell membranes. So we have a hydrophilic head at the top, and then hydrophobic tails. One tail is always saturated, and the other tail is an unsaturated fat. They look kind of like a bobby pin. Okay, good. So we've got, if you were to put these phospholipids into water, all on their own, they would form these micelles, these little bubbles where the outside of these little balls of phospholipids would be hydrophilic on the outside, and all the hydrophobic tails would be pointed to the center. To make a membrane is where we have two layers together. So there's an outside layer that's all hydrophilic heads. All of the hydrophobic tails point together in the center, and then the inside layer of the membrane is also hydrophilic heads. All right, how do the structure of phospholipids contribute to the structure of cell membranes? So I showed you just a straight chain of that. Imagine it makes a whole sphere to encompass a whole cell. Describe how important that might be here, or draw a picture. All right, so this is a nice little summary table of the 4 different types of macromolecules: sugars or carbohydrates, lipids, proteins, and nucleic acids. Here's examples of their monomers, examples of their polymers, and the function that they serve in living cells. You should be familiar with all this information. You'll see it again on the test. All right, so how do fats function as an energy storage molecule? Pause, write your thoughts here, and then continue. Why do phospholipids form membranes while triglycerides form insoluble droplets? What's different about the structure of a phospholipid compared to a triglyceride fat? All right, so by now, you should have an understanding of the basic chemistry of life. You should be able to list the 4 major groups of organic molecules associated with living things. You should be able to describe the general function of each major group of macromolecules. You should be able to explain factors that affect enzyme activity and explain the difference between dehydration synthesis and hydrolysis. All right, if you have any questions, please write them down, take them to student office hours so we can chat about them, or bring them to class, all right?